Hi everybody, in this video we're going to learn about translation in bacteria. So to start with, just a quick terminology overview to make sure that everyone is on the same page. So this is a diagram of a bacterial ribosome. Hopefully you remember that the small subunit is the 30S size. The large subunit is the 50S size. The ribosome is reading the messenger RNA, and the kind of language, if you will, on the messenger RNA is in sets of 3 nucleotides. These sets of 3 nucleotides on the messenger RNA are called codons. These codons will match with the complementary anticodon, a set of 3 base pairs on the transfer or tRNA. tRNAs will enter the ribosome carrying an amino acid, and so a tRNA with an amino acid is called a charged tRNA. These amino acids are going to be moved as the translation happens, and the bond that will form between them is called a peptide bond. I know that you can't really see these because the tRNAs are on top, but so the large subunit of the ribosome holds 3 transfer RNAs, and each transfer RNA goes into like a little kind of cup spot where it will sit. And so the first spot where the tRNA comes in is called the A site or the acceptor site. The middle spot is called the P or peptide site, and then the third spot is called the E or exit site. And so we will be referring to these again later. So hopefully this gets you guys all kind of caught up on terminology for translation, which is making proteins. So the first thing that we actually need to talk about is how we make the charged tRNAs. And so charged tRNAs, remember, are tRNAs with amino acids stuck onto them. The fancy name for charged tRNAs is aminoacyl tRNAs. And so the reason that I mention that is the enzyme that builds or assembles these is called aminoacyl tRNA synthetase. Hopefully, as soon as you see this T right here, and you see that this is synthetase, you will remember that synthetases use ATP. So the aminoacyl tRNA synthetase is this enzyme here, and so it takes the amino acid, the ATP, and the tRNA and sticks the amino acid on top of the tRNA. There is one of these types of enzymes for each amino acid. And so even if there is more than one codon for that amino acid, there is only going to be one aminoacyl tRNA synthetase per amino acid. And so this enzyme will use ATP to assemble the charged tRNA, the tRNA with the amino acid. The reason that this is important is, do you guys remember before when I told you that the squiggle bond indicates high energy bond? So the energy in the ATP has actually been transferred into this bond right here between the tRNA and the amino acid. And so this is the energy that is going to be used to form the peptide bond during actual protein synthesis. And so the energy originates with ATP, is stored in this bond here on the charged tRNA. Another thing that you might see is, do you guys remember when we talked about making polysaccharides and how glucose with a UDP was called activated glucose? So similarly, an amino acid with the energy from ATP can also be called an activated amino acid. So it's a charged tRNA, also called an aminoacyl tRNA, but the amino acid can be called an activated amino acid. This process is done by aminoacyl tRNA synthetase, of which there is one per amino acid, And this is an important process because it transfers the energy from ATP to be stored between the tRNA and the amino acid, and it is this energy that will ultimately power the formation of the peptide bond. So getting the right amino acid onto the right tRNA is really an important part of successfully making proteins, and so that's why we're learning about this. Okay, you will either be super excited or super frustrated when I tell you that the 3 steps in translation are initiation, elongation, and termination. So again, I would not really rely very heavily on the names of those steps because they are the same step names for every single process, and I don't want you guys to get jumbled. Just like before, initiation is the assembly of all of the things that we need. Elongation will be the making the protein, and then termination will be the disassembly of all the things. And so initiation will set up something called the initiation complex, and I have covered up part of it for just a second so that it's not so much going on. And so the first thing that happens is the 30S, or small subunit of the ribosome, will interact with the messenger RNA. There are some small helper proteins involved in this process, and the ones that help with initiation are called the initiation factors. And you see this little one right here, initiation factor 3. Initiation factor 3 will prevent the large subunit from binding until the small subunit has acquired the messenger RNA and the first transfer RNA, which is what is happening underneath this paperclip here. And so here is our first tRNA. Because it's the first one, it is called the initiator tRNA. And so the initiator tRNA will bind to the start codon. We'll get back to that in just a second. So the initiator tRNA will bind to the start codon. This is helped into place by yet another initiation factor, IF2, and the ribonucleotide energy source, GTP. And so this initiator tRNA will come, the messenger RNA will come, the small subunit of the ribosome will come, and all of this together is called the 30S initiation complex. So a couple of side note things that I need to tell you. The first one is that you are used to the start codon being AUG. The start codon is always AUG in eukaryotes. The start codon is not always AUG in bacteria. The start codon is frequently AUG in bacteria a lot of the time, but not 100% of the time. The other extremely bizarre thing is you will see that this amino acid has this very mysterious F in front of it. So in bacteria, when the start codon is AUG, then the first amino acid, the amino acid on the initiator tRNA, is actually something called formal methionine. Often, the formal group will be cut off, and so you'll end up with just regular methionine in the finished protein. But it is noteworthy that bacteria use formal methionine, abbreviated FMET, to go with the codon AUG instead of using just regular methionine. These other two, GUG and UUG, are alternate start codons that are not uncommonly used in bacteria. So AUG is still going to be the most common start codon, but these will be other start codons that are used often enough to make note of them, GUG and UUG. And so the 30S subunit, the initiation factors are helping. You are not going to need to keep track of which initiation factor is which. Just know that there are small proteins called initiation factors that help with this. And then the messenger RNA comes in. Okay, so one of the other things that we have to figure out is, so here in this diagram on the messenger RNA is this set of 3 bases. So we know that 3 bases on the messenger RNA is the codon. But what is it about this particular AUG that tells the cell that it's the first AUG, right? So it's 100% possible that there could be other methionines somewhere else in the protein. So what is special about this particular AUG that the cell can know that it's supposed to be the first one? And so this very front part of the messenger RNA, it's not really well shown in this picture, but actually like hooks around the bottom of this, and this is the 16S ribosomal RNA. So you guys remember when we talked about using the 16S ribosomal RNA for identification of bacteria? So it's actually the 16S ribosomal RNA will interact with a section on the ribosome called the Shine-Dalgarno site. These are capitalized because it was discovered by 2 Australians, and these are their names, and so it's going to be capitalized. You might see it sometimes as the SD site, and it is also called the RBS site because it is the section on the messenger RNA that is going to bind the ribosome. So the ribosome binding site, also called the Shine-Dalgarno site, is a piece upstream or ahead of the start codon, and this is actually what is going to wrap around, attached to the 16S ribosomal RNA in the small subunit, and it is this interaction between the Shine-Dalgarno site and the small subunit that positions the start codon in the right place to be the first codon. Okay, so all of this together makes the 30S initiation complex. These are the 3 things that comprise the 30S initiation complex, and these are required to set the complex up. Alrighty, so here is a slightly better picture where you can see the ribosome binding site kind of bending around and interacting with the small subunit of the ribosome. This is like more dark than I expected. So what I wanted to show you guys, I don't know how well you can see this, but I really want you to see it, so I'm going to see if I can zoom in more. Okay, so I thought this was going to be pink instead of this kind of purple color, but whatever. And so here is a methionine. I don't know if you guys can see that, but that right there is an M, which stands for methionine. And so if we look in this top sequence of the DNA, do you guys see that? We see ATG. And so when we see ATG, we know that that is the DNA version of AUG. And so that should immediately tell us, if I write over here, can you guys see? Nope. That this is the coding strand, because this strand looks to us like messenger RNA. This other strand right down here is actually the template strand. So it is the bottom strand that was actually read by the RNA polymerase, but the messenger RNA produced is going to look like the coding strand. And so this would be where the start codon is, the AUG in the messenger RNA, which will give methionine. And so the ribosome binding sequence, or the Shine-Dalgarno sequence, is going to be, let me zoom out a little bit so you guys can see this better, is going to be a high AG region. So purines are nitrogenous bases with 2 rings in them, and so A's and G's are those. Despite being the short word, I don't even know they have more rings, nevertheless. And so the Shine-Dalgarno sequence is going to be a high AG sequence that is about 10 bases upstream of the start. And so let me zoom back in so that I can show you. I just think this is the neatest thing, because you find these so often and they're just patterns hidden in the DNA. So if we go from the start, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 is the first G in our Shine-Dalgarno sequence, and they can be between 3 and 10 bases long. Basically, you just look for a high AG concentration that is 10 base pairs up from your start, and there's going to be your Shine-Dalgarno sequence. And so this is how computers can predict the starts of genes, is they look for every possible ATG in the DNA because it's secretly indicating AUGs, and then they check, is there a good Shine-Dalgarno 10 bases up? Is there a good Shine-Dalgarno 10 bases up? And then the computer will say, we predict the start will be this methionine and not this methionine, because the Shine-Dalgarno would be 13 away from that A, and that's too far. So anyway, I just think this is the neatest thing. Whoopsie, got super excited and whacked to the papers. So the Shine-Dalgarno sequence is on the front of the messenger RNA, and is how the mRNA is positioned on the small subunit of the ribosome in order for the initiation complex to form. Alrighty. So once we have together the 30S initiation complex, which again is the small subunit, the initiator tRNA with the formal methionine and the messenger RNA, that is when the 50S subunit will come on. And so, so you might remember initiation factor 3 prevented the 50S. In this assembly process, initiation factor 1 comes on and 3 is kicked out. And so once the mRNA comes in, then the 30S, then the 50S, the large subunit, is allowed to bind. And so the 50S subunit will come into place. This requires still the use of initiation factor 1 and more GTP. And we get the 70S initiation complex. So the complete ribosome with the messenger RNA and the initial tRNA. You can see that this first tRNA ends up in the P or peptide site of the ribosome, which is the middle site. So this is going to be important here in a second. I have highlighted in pink every time that GTP shows up. For a couple of reasons. Number one, because it's not ATP, and I want you to be aware that there are other energy sources in the cell besides ATP. But number two, because it takes a freakishly large amount of GTP to make proteins. And so this is why bacteria will often use carbohydrate food sources before they will use protein food sources, because proteins are energetically very expensive for the cell to build. So this is the 70S initiation complex, and now the cell is ready to start making protein. So this is a summary initiation slide, which you can read in the PowerPoint because it's kind of small printing. We're going to move on. Okay, so I have this summary sheet thing that has all 3 steps. So I'm just going to show you one step at a time. So initiation is the assembly of all of the parts needed to make protein, which are listed here. As a reminder, there are 3 different options actually for start codons in bacteria, although AUG is still the most common one. These are the other amino acids that are encoded, in case you were wondering. Okay, so once initiation is finished, we are ready to do elongation, or the actual making of the protein. Okay, so you might remember, can I zoom in? Of course I can, I have the thing. Let me scooch this up some. I'm sorry that I made this picture so weensy because now it's the one that I want. Anyway, so at the end of initiation, then the initiator tRNA was in the P site, right? So the new tRNA carrying the new amino acid comes into the A site. It is helped into place by something called elongation factors. This giant blue globby thing is an elongation factor. Right, so you've by now figured out that the name of the protein is just the name of the stage and the word factor. This process also GTP to bring the tRNA into place. And so here is the GTP being used here. Okay, so on Moodle also, there is an excellent animation that you should definitely watch because it is much smoother than I am at this part. But I think this is super nifty, and so I'm going to show you it anyway with my awesome paperclip Play-Doh puppet situation. But you should still watch the animation. Whoopsie-daisy. Alrighty, so here is going to be our slidey messenger RNA. I made these beforehand. So here is our tRNA in the P site, and so it's got some amino acids. Here comes new tRNA into the A site. Ta-da! And so in something called the peptidyl transferase reaction, is that all on? Peptidyl transferase reaction. The amino acids from the P site tRNA are moved onto the A site tRNA. So the bond that is formed here is between the amino acids is the peptide bond. And since the amino acids are transferred and we form the peptide bond, hence the peptidyl transferase reaction. Okay, so the ribosome actually moves, but I did not figure out how to do that with the paper. And so we're going to pretend like the ribosome stays the same, and we're going to move everything else. Oh, hang on. See if I can stick that together better. Okay, so the ribosome moves, and so we're going to slide it down. So like this. And so now the previous P tRNA, which is now empty, is in the E4 exit site, and the one that was in the A site that accumulated all of the amino acids is now in the P site. Okay, so now we are ready, you guessed it, for yet another tRNA to come and enter the A or acceptor site. At this point, unsurprisingly, the peptidyl transferase reaction happens, and all of these amino acids get moved over to here. A new peptide bond is formed, the ribosome scooches down, etc. So that is the whole process of elongation. That's it right there in a nutshell. Do you guys remember at the beginning of this when I spent all of that time talking to you about the aminoacyl tRNAs and how the energy from ATP got put into that bond right there? This, again, is the energy that is used to synthesize the new peptide bond. And do you guys remember the very beginning of the semester when we learned about ribozymes? And I made you memorize that the most common ribozyme was the ribosome. This is when we use that right now. I know that you have secretly been wondering this whole time. It has finally happened. And so it is the 23S ribosomal RNA, which is the catalytic RNA in the large subunit right here, that actually catalyzes the peptidyl transferase reaction. And it is this reaction that moves the amino acids and forms the new peptide bond. And then the next charged tRNA can come in. So every time a tRNA comes in, per this picture, I'm just going to zoom back in on this fast so that you can see it. So every time a new transfer tRNA comes in, it requires an elongation factor escort, and it requires GTP. So we require GTP to assemble the ribosome. Now, every single time we bring a tRNA in, we require GTP. So still more GTP is being used. Okay, so that is the making of the peptide bond in the peptidyl transferase reaction. And then, like I said, the ribosome, I guess I could have just showed you this page. The ribosome scooches down, which is called translocation. and then unsurprisingly, GTP is used for the scooching down process. And so then the now empty tRNA is in the E site and it goes out. A brand new tRNA with a new amino acid will come into the A site, and this requires elongation factors and GTP. And so pretty much every single time anything happens in translation, GTP is required. So this is just an incredibly energy intensive process. Okay, so like I said, the E site tRNA is ejected. The ribosome will scooch down, which is called translocation. And then a new tRNA will be brought in. This requires more GTP. So the cell keeps doing this, or the ribosome keeps doing this, until a stop codon is reached. So you guys hopefully remember that there are 3 stop codons. These do not have matching tRNAs. So I'm supposed to show you the next part of this page now. Okay, so elongation, which is the translocation or the scooching of the ribosome, is how new amino acids get added. This happens in the peptidyl transferase reaction, which builds new peptide bonds. This peptidyl transferase reaction is catalyzed by the 23S ribosomal RNA, which is a ribozyme. And again, just as a reminder, this requires elongation factors and GTP and depends on the correct match of the 3 bases in the mRNA called the codon and the 3 bases in the tRNA called the anticodon. Okay, so once we get totally finished with this, then eventually the ribosome will go down until it reaches a stop codon, of which there are 3. These are them here. And so the stop codon does not code for a tRNA to come in. It codes for a separate protein called a release factor. And so the release factor will come in, and it's not really shown in this picture, but actually I guess there's several of them. Release factors will come in, and the release factor will cut off the protein and allow the ribosome to disassemble. Unsurprisingly, this requires more GTP. So as soon as the ribosome disassembles, initiation factor 3 will come on, and it attaches to the end of the small subunit. And so you guys might remember what initiation factor 3 does is prevent the large subunit from joining the small subunit until the messenger RNA and the initiator tRNA are in place. And so as soon as the ribosome is disassembled, initiation factor 3 will join. Trying to see through my page here so I can see what's next. Oh, this is just a lovely overview slide. Ta-da! Yeah, you can read this more later. So here's the rest of my summary page. So in termination, the ribosome disassembles. The stop codon codes for the release factor. GTP is also involved in this process. And as the ribosome disassembles, initiation factor 3 will join the 30S small subunit to prevent the large subunit from reattaching. And then we just start back over. So one thing, actually kind of randomly, that I would like to show you is this picture here makes it look like the messenger RNA is just floating about in the cell. But hopefully, since we've talked a lot about transcription and translation being simultaneous, you will realize that this is not a real thing. And so really, the RNA polymerase is like globbed on the end somewhere, and the ribosome will just come and attach while the messenger RNA is still being produced, actually. So yeah, do not be misled by that. Okay, so here we go. So we have a couple of quality control questions that we need to ask. How can we make sure that the correct amino acid is added into the protein? This is really important because if the wrong amino acid is added, the protein will not fold correctly, and the protein function depends on its having the correct shape. And so we need to be really confident that the correct amino acid is put into the protein. And so there are a couple of places that this type of quality control happens. The first is with the attaching of the amino acid to the transfer RNA to begin with. And as a reminder, this is done by the aminoacyl tRNA synthetase, which, as a reminder, requires ATP. There is one synthetase for every amino acid, and all of the tRNAs that will match that amino acid will fit into that same synthetase. And so this enzyme is responsible, this type of enzyme is responsible for making sure that the correct amino acid is matched with the correct anticodon. The second step is making sure that the correct anticodon gets matched with the correct codon. And so there are a couple of special terms here which you might have heard before: degeneracy of the code and wobble. So degeneracy of the code, degenerate code is basically like a redundant code. And so this is the term that describes the fact that for most amino acids, there is more than one codon that will code for that amino acid. What this means is that if there is a mutation, an inherited change in the DNA, but the mutation changes it to one of these other codons, the protein will still be assembled and work normally. And so degeneracy refers to the fact that its degeneracy is like redundancy, and refers to the fact that there are multiple codons for each amino acid except for methionine that only has one, but all of the other amino acids will have multiple codons. Wobble is the phenomenon whereupon, let me see if I can find a picture to show you guys here. Okay. All right. So this is the codon-anticodon match right here, right? And so these three bases are the anticodon on the tRNA. Let me look for a different color here, just a second. And then these three bases on the mRNA are the codon, okay? And so they draw it like all of them exactly match up. But really, this first set matches the most strongly. It is the most important matchup in the codon-anticodon match. This second set matches pretty strongly, not as strongly as the first set. The third set, kind of matches. It's okay if it doesn't, etc. And so wobble is the idea. Whoopsie, sorry, I forgot to zoom back out. So wobble, I'm trying to get this to be straight. I'm in a struggle. The Elmo is crooked, I have discovered, and so I have to like hold the paper crooked in order to make it look straight, and I'm failing at this job. Okay. So wobble is the phenomenon whereupon, because the third base of the codon-anticodon match actually does not interact as strongly as the first base or the second base, if the third base does not match, then the tRNA will still join with the mRNA, and you will not. So this will be like a silent mutation. If there has been an inherited change to the DNA that changes the third base, it won't matter, because wobble allows a tRNA to match even if the third base of the pair of the triplet does not match. So wobble has to do with the third base not matching. Okay, so this slide is to show you, and again, you can look at this more on the uploaded slides, but this slide lists for you each of these yellow boxes is a different commercial antibiotic that works on protein synthesis in bacteria. And so this slide explains to you how these different antibiotics work. Just again, as a slight reminder, bacteria and mitochondria are basically analogous. And so mitochondria also have ribosomes that are making proteins. Mitochondria also use formal methionine, FMET, as their first amino acid to match with AUG. And so if you take one of these antibiotics that works on the bacterial ribosome, you might experience some negative side effects if it impacts a lot of your mitochondria. Excuse me, a few of your mitochondria can stop producing proteins, and it's fine because you are huge, much bigger than the bacteria, so they will die before you do. But you might still get some negative side effects, and so especially with, I think, some of the tetracyclines. So just do be aware that that is why you can get negative side effects if the antibiotic works on the ribosome, is because mitochondria also have ribosomes. So, okay, so you guys remember when I told you that bacteria mostly do not have introns because transcription and translation are simultaneous? Bacteria, weirdly enough, do have things called inteins. I don't even know. So inteins are disruptive parts inside of proteins, which will self-splice out. I don't know very much about these, and I cannot give you an example of them. I just read that they existed and wanted you to know so that you would be in the loop. If someone says to you something about inteins, you will know that they are not just making up stuff. So there you go. Once the protein is produced, it will be folded. So most proteins will self-assemble using different chemical interactions as they are being made. And so, yeah, usually protein assembly happens with no problem on its own. In some cases, the protein might need help folding correctly. And in those cases, then they use proteins which have up to 4 different names. So these proteins can be called molecular chaperones, but they can also be called chaperone proteins, or they can be called chaperones, or they can be called heat shock proteins. And so heat shock proteins will help to refold misfolded or unfolded other proteins, actually in all different kinds of circumstances. So if you have a drought, heat shock proteins will work. If you get too hot, heat shock proteins will work. If you get too cold, heat shock proteins will work. So heat shock proteins were first discovered when they like flash heated E. coli, like quickly heated it to 70 degrees and then quickly cooled it back off so that it didn't die, but like the proteins went all weird. That is how they discovered these chaperone or heat shock proteins that will help to refold existing cellular proteins. Later on, they realized that these are not just for heat. They're like general stress response proteins in bacteria. And so just if you hear about them other places, they're not only for heat. Each of these molecular chaperone proteins is powered by ATP. They work in pairs, and examples of famous pairs are DNA J and K and GRO EL and ES. So we are not going to talk anymore about how these work. They are amazing and cool, and they make these little kind of barrels with hats on them. But that's all we're going to say. So the last thing that we need to talk about is how proteins get out of the cytoplasm. And so these proteins might be headed for the membrane, and they might just stay embedded in the membrane, or they might be headed for export out of the cell altogether. There are actually several different ways that this can happen, but we are only going to learn the 2 most common export systems, and that is the SEC system and the TAT system. And so you can see here the SEC system is primarily for not primarily, it's totally for unfolded proteins, and these proteins could be headed out of the cell or they could go and just stay in the membrane. But in either case, they are exported in the unfolded state, and then they will fold whenever they get to the place that they're going. The TAT system is the system which exports folded proteins, and these proteins are targeted for export by the presence of 2 arginines in the signal sequence, which is going to be at the very beginning of the protein. And the TAT system is less common. The SEC system is super, super common. The TAT system is a little bit less common, but it's used for folded proteins and requires this special signal sequence. So that is protein production, aka translation, in bacteria, and secretly also in mitochondria.